Chapter 8 is on microbial genetics. This is a pretty big chapter, so what I've done is divide it up into several units just to make it a little more manageable. This first section is on genetics and DNA replication. Genetics is the study of heredity. So this is passing genetic information down to offspring. This is what we call vertical gene transfer. And this is what we talk about with both bacteria and eukaryotic cells, humans. The way that genes are passed on is to the next generation. A gene is going to be a certain segment of the DNA that codes for a protein. We would not be able to make proteins without the code in the DNA, and that code for the protein would be kept on a gene. Now, we know that prokaryotic cells are different than eukaryotic cells. They have a circular chromosome, so they don't have linear chromosomes. Humans have 46 chromosomes, or 23 pairs. They have a circular chromosome, and some bacteria can have plasmids. Plasmids contain useful genes. I call them nice to have genes or accessory genes. They are not going to contain necessary information for the survival of that bacteria. That's going to be contained on that circular chromosome. So not all bacteria will have plasmids. We'll talk a little bit more about plasmids because many of these plasmids can contain antibiotic resistant genes, meaning that if that bacteria has a resistant gene for an antibiotic, it can actually live in the presence of that antibiotic so it doesn't kill it. But not all bacteria will have plasmids. So just a little review on DNA. DNA is a biomolecule. It is a large biomolecule. It is a nucleic acid. Like all polymers, it's made up of monomers. And the monomers of nucleotide or nucleic acids are nucleotides. And so a nucleotide has a sugar, a phosphate, and a nitrogenous base. So in the case of DNA, the sugar is deoxyribose, and this is how DNA gets its name, deoxyribonucleic acid. Another nucleic acid that we're going to talk about in protein synthesis is RNA. RNA is single-stranded. It's not double-stranded like DNA. And the sugar is a little different. The sugar is ribose. So again, that's how RNA gets its name, ribonucleic acid. So on that little picture on the bottom, you can see on the right side where that DNA is in that double helix form. It almost looks like a slinky or like a spiral staircase. If we were to actually stretch it out and make it look like a ladder, on the outside part of the ladder, you would see the sugar and the phosphates. That's what we call a sugar phosphate backbone. And then on the inside, this is where we have the base pairing. So our nitrogenous bases are going to pair and we're going to have a match or its complement. So if you take a look on the one side, we have an A. Adenine pairs with thymine. The way that I remember that is if you look at the hard angles of the A, so there you can see that hard angle of the A, and the T, they have sort of like those hard angles, whereas the G and C have sort of those rounded shapes. So that's how I remember that guanine and cytosine go together and adenine and thymine go together. So if you see an adenine on one side, you're going to see a thymine on the other side. If you see a cytosine, a C on one side, you're going to see a G on the other side. So that is how we would actually get our complement of DNA. So looking at that code, A, C, T, G, on the one side of the DNA, on the complement side of the DNA, we would see the code TGAC. So in DNA, A pairs with T, G pairs with C. Now, when we get to protein synthesis and we talk about RNA, there's actually a base substitution. there's no thymine in RNA. There's a base substitution of uracil. So there's actually a U. And so I always tell this sort of Valentine's Day riddle. I'd rather be RNA than DNA because RNA has U. I actually got to say this on Valentine's Day, and I was really excited. The entire class wasn't as excited as I was, but I thought it was pretty cool. So just to let you know, if you see U's, you always know that we're in the language of RNA. If you see T, you know that we're in the language of DNA. So a little bit more detail of the DNA. So on the right side, you can see that double helix. And on the left side, this is when we stretch it out as this ladder. So on the outside part of the ladder, you can see that sugar phosphate. On the inside part, you can see those nitrogenous bases. So every place you see a T, on the other side, you're going to see an A. Every place you see a C, you're going to see a G on the other side. You'll notice if you look at this picture where the C and the G and the T and the A are connected, you'll see these dotted lines. Those are actually hydrogen bonds. Hydrogen bonds, I hate the term bonds because they're more like attractions. They're not really sharing electrons. It's just that those molecules are attached and attracted to each other. So that's what you're seeing that's kind of holding those together are those hydrogen bonds. Now, the other thing that you're going to see about this picture is if you take a look at the left side, it's got 5' to 3', which is facing down. And then on the right side, it's got an arrow that's facing up that says 5' to 3'. So what on earth does this 5' to 3' mean? Well, if you notice on the top, if you look at that D, there is actually that sugar, right? And you can see at each of the corners, there's a 1', 2', 3', and so forth. Those are actually carbons. And so this is how biochemists actually label the carbons. So if you see that 5 right there, that 5', that's that 5' facing up. If you look at the bottom of the DNA and you see that 3', that 3' is facing down. So that's why the arrow is going 5' to 3'. If you look at the other side of the molecule, we've got our 5' carbon, which is facing down, and our 3' carbon, which is facing up. So you will notice that the arrow is going up in that 5' to 3' direction. So what this means is that DNA is actually running in opposite directions. This is what we call anti-parallel configuration. So that means that DNA is running in opposite directions, kind of like two trains with two train tracks next to each other, where they're actually going to be going in different directions. One's going north and one's going south. All right, why is this a big deal? This is a big deal because as we start to get into how DNA is made, how it's synthesized, the enzyme that makes DNA is DNA polymerase. DNA polymerase can only add a nucleotide in a 5' to 3' direction. So what that means is that the two sides of DNA are actually made a little differently. And we're going to get into that as we get into the details. So one of the things that DNA, of course, has are genes, and genes are going to code for proteins. Now, with humans, we have two different alleles, and an allele is a version of a gene. And that particular gene is going to be at a location on the DNA. That's what we call a locus. So with humans, we have two alleles. And an allele is a version of a gene like if we had free earlobes or attached earlobes, or what our blood type is, if we're type O, if we're type AB, if we have brown eyes or blue eyes, although eye color is a little more complicated as far as heredity. Prokaryotic cells only have one allele because remember, they are going to reproduce by way of asexual reproduction through binary fission. So we have two alleles because we get one allele from our mother and one allele from our father. Prokaryotic cells do not have a lot of genetic diversity because they only have one allele. Now, one of the things that we talk about with DNA are mutations. What are mutations? Mutations are changes, or sometimes we call them mistakes in the DNA sequence. And so if mistakes happen, they can actually be passed on to the next generation. I often call mutations kind of the good, the bad, and the indifferent. I kind of take that from the old Clint Eastwood movie, The Good, the Bad, and the Ugly. Sometimes mutations are good. So if a mutation arises where now a bacteria is resistant to an antibiotic, good news for the bacteria, maybe not so good news for us. They can be bad, where they can cause a protein not to fold correctly, so it can't function. That's going to be bad. Or it could be indifferent, where even though we change the DNA, we're not changing that protein. We haven't changed the phenotype, the outward appearance, and it doesn't really make a difference. And we'll talk a little bit more on mutations at the end of this chapter. All right, so what does DNA do? DNA is extremely important. We would not be able to make proteins without the DNA. Our DNA has all of the genes to code for all of the proteins that our body needs. It defines who we are and what our different traits are. So first off, before a cell divides, and whether we're talking about a human cell that's dividing by mitosis or a bacterial cell which is dividing by way of binary fission, we have to synthesize that DNA. We have to replicate it. Why? Because if that cell divides and it hasn't replicated its DNA, it's going to have half the amount of DNA and it's not going to be able to function. The other thing the cell needs DNA for is to make proteins. And one part of this chapter, we'll be getting into protein synthesis. Protein synthesis, if you remember learning about it before, is actually a two-step process. There's transcription, making a copy of the gene, and then translation, taking that copy and then turning it into that protein, something three-dimensional that that cell can actually use. So we're going to be going through the process of that DNA replication. So DNA replication is what we call semi-conservative replication. If you've ever tried to do a project, if you've seen something online and you're like, you know what, I really want to replicate that. Like if you've seen something on Pinterest, it doesn't always go as planned. One of my favorite things to look at on social media is different cakes and how people try to replicate it. So they usually show a picture of what the cake should look like. And then they have a picture of what it actually looked like, and they're kind of hilarious. The other thing I like is that if somebody goes to buy something, like this is what the picture said they were getting, and then this is what they actually got. In DNA replication, we don't want a Pinterest fail. We don't want an Amazon delivery fail, because that means that that cell is not going to be able to function. So nature has found a way to minimize mutations, and that is through semi-conservative replication. So rather than taking a strand of DNA and making two brand new strands, nature has found a way to minimize that mutation and mistakes that could happen by conserving half of that original strand. So if you take a look at this picture, here we've got the original DNA in blue. Through the process of semi-conservative replication, that DNA is separated and then new DNA is built on the other side. That's the pink DNA. So that original strand is used as a template, and then we've got the new DNA built on the other side. That's going to help to minimize those mutations. As we start to get into the process of DNA replication, the enzyme that builds the new DNA is DNA polymerase. And DNA polymerase has some proofreading capability. So if it goes to add a nucleotide, when it adds it and it doesn't quite match, it can actually substitute in the right one. So it has a little bit of proofreading capability. So there is some baked in minimizing those mutations, not only with DNA polymerase being able to kind of proofread, but in this semi-conservative replication. Because the last thing the cell needs is a Pinterest fail. We certainly don't want that, because then the cell is not going to be able to make its proteins. So we're going to go through the process of DNA replication. So what we looked at before is that DNA runs in that anti-parallel configuration, opposite directions, that 5' to 3'. DNA polymerase can only add those nucleotides in a 5' to 3' direction. Where new DNA synthesis happens is a replication fork. So DNA is that double helix. We're going to have to open up that DNA, and that is going to be the process of an enzyme. So I'm going to attempt to draw a strand of DNA right here. So here's our DNA right here, not a very good strand of DNA. And we need to actually open it up a little bit. So I'm going to erase this part, and we're going to open up our DNA right here. If I can actually draw this DNA. So we're going to open it up right here. And this is where new DNA synthesis is going to happen. So we have an enzyme, helicase, which is actually going to unwind that DNA. Then, once it's open, and let me choose another color here. Now what we're going to do is there's another enzyme which is going to put down a little piece of RNA. This is an RNA primer. There's another enzyme, primase, which actually puts this down. And this is going to be kind of like the start here. This is where that enzyme, DNA polymerase, and let me get another color for that, is actually going to jump on. So DNA polymerase is going to recognize that, and it's going to jump onto that DNA. Now it's actually going to build the DNA on the other side. So if we go back to our original color here, if we have a C, if we have a C, what we're going to have on the other side, and this pen isn't drawing very well, we're going to have a G. If we have an A, then we're going to have a T. And my pen is not drawing very well. So we're going to be seeing a more detailed picture with this, which is actually coming up. And we will talk about the enzymes and why DNA is built a little differently on both sides. So it's a little easier to see from this picture right here. And it's a little difficult to kind of write on this screen here. But here we've got our DNA. So you notice that that DNA is opening up. The enzyme that's going to help us to do that is helicase. So we're going to open up that DNA, and you see where it kind of branches off, almost like a two-tine fork. That's what we call a replication fork. That's where new DNA synthesis is going to happen. So the one side is the 5' to 3' direction. So you notice that that side is in blue, so that's on the bottom. This is what we call our leading strand. Because of that chemistry, that DNA polymerase is actually able to add those bases continuously. So the leading strand is made in one straight shot. On the other side, because we're running in that anti-parallel configuration, the pink side, it's going to have to be made in pieces. This is what we call the lagging strand, because several RNA primers need to be put down so that RNA polymerase can add that chemistry one nucleotide at a time with that 5' to 3' direction. So this is what we call the lagging strand. Eventually, another DNA polymerase is going to come in. It's going to fill in those gaps. And if you take a look at those bottom pictures, this is where we have the half of the original strand and half of the new DNA strand. So at the end of DNA replication, each of those two strands contains half original DNA and half new DNA. Because of the chemistry and the way that DNA polymerase can build one nucleotide at a time in that 5' to 3' direction, it's made a little differently on both sides. So on one side, it's made continuously. That's what's called the leading strand. The other side, it's made in pieces. That's called the lagging strand. Those pieces we name Okazaki fragments for the scientist that actually discovered it. So this helps to minimize those mutations. Now, I know it's a little difficult to kind of see from a two-dimensional picture, and it was a little difficult to draw on the screen. So I did post several videos. So there is one video that's a couple-minute video. I think it really does the best job of describing and showing all of those different enzymes and how DNA polymerase works. And it shows you that semi-conservative replication. Amoeba Sisters Crash Course also has protein synthesis videos. If you're like me, where you need to see all of these moving parts, I'm really going to encourage you to look at those videos to kind of help you get that understanding. The other thing that you want to do is to describe it. I always say that if you can explain it, then you know it. So if you can go through that process and talk about how the leading strand and lagging strand are synthesized a little differently, then you're going to be in good shape.